This is another presentation for the recombinant BRCA1 gene cancer, which is also known as breast cancer susceptibility gene 1, using the vector PC DNA 3.1 in the mammalian cell of HEC2932. We want to talk about breast cancer. Breast cancer is found in the mammalian gland or duct of the human, which appears mostly in the upper or outer quadrant of the breast. The breast cancer gene, which is the BRCA1 gene, is a tumor suppressor or a caretaker gene, which is responsible for repairing DNA and also chromosomal damages, with an important role in the error-free repair of DNA double-stranded breaks. The reason for the expression of this protein is to study and identify a potential drugs to treat cancerous genes like breast cancer. So the next slide talks about the vector. The vector or plasmid used for this expression is a pcDNA 3.1 vector, which is a vehicle used to carry our gene of interest, which is our HEC293 cell. Since we are using the mammalian virus, the PCMV promoter in this vector circles a megavirus, which causes a large amount of transcription to occur, and it produces large amount of mRNA. There is also a poly-A tail, which is the BGHPA, which in every promoter of a vector it promotes a bacteria on the mammalian expression of vector that is needed to correct the mRNA that will be expressed inside our mammalian cell. We also have an F1 origin. This one is also a replication or copying and making simple stranded DNAs, and there is also an ampicillin, which emphasizes on resistance so that we can work with the plasmid in the cells. There is a myrcene, which produces a gene that confers resistance to the drugs. This pcDNA 3.1 vector is widely used and designed for high-level stable expression in mammalian cells. There is a restriction enzyme which we use, which are XHO1 and KPN1 to digest our plasmid at the cloning site, and we do the same thing in our gene with our gene of interest, which is the BRCA1 gene. So we introduce this gene into the plasmid, which is also the vector, the pcDNA 3.1, with a T4DNA ligase. That means that means the ligation process starts. We take our DNA plasmid that contains our eukaryotic gene and incubate the solution to the cell for 10 to 30 minutes. This allows proper ligation to take place. We're talking about the transformation of our mammalian cell, which is a HEC293T. The transformation of our mammalian cell, which is the transformation of the recombinant DNA plasmid into the mammalian cell, which is done by the heat shock. And the solution is placed on the water bed for 42 degrees Celsius for 45 minutes, and is also placed in the ice for 2 minutes. Then we add our recovering medium that improves the molecular uptake, which sterilizes the cells, which allows the bacterial cells to pick up the recombinant plasmid. There is an incubation of the culture at 37 degrees Celsius for 45 minutes to 1 hour. Then we transfer our bacteria on an antibiotic plate, which is the IBGT medium containing our ampicillin, knowing that our vector has the ampicillin resistant gene. The bacteria placed on the antibiotics plate will distinguish the ones that has the ampicillin resistance and the ones that doesn't. The one that doesn't have the ampicillin resistance will die, while the ones that have ampicillin resistance will propagate, going to form colony. So introduction of this IBGT to the BRCA1 protein to carry out extraction of the ligase of this cell, where the vector has the his tag that binds to the gene of interest, which is our HEC293T gene, and it will be purified using affinity chromatography. We also use Sanger dioxy sequences to purify our proteins. We then go to the transfection of the HEC293 gene. How to transfer the cell into the plasmid? The HEC293 G cells is a cultured dish at the approximate density to ensure that we reach 60 to 80% confluence of the day on the day of the transfection. Remove the growth medium from the cells and replace it with optin-mem. Then we add the DNA transfection reagent, which complexes downwise of the cells. We dilute the vector with the gene of interest and insert our optimum medium. Then we optimize the DNA concentration based on the transfection reagent recommendation. We take a separate tube, dilute the lipofectamine in the optim medium, then incubate for 10 minutes at room temperature. When we incubate the cell, for roughly 18 to 24 hours to allow the gene expression. After that, for 48 to 72 hours, we analyze the transfection efficiency by fluorescence microscopy. This is all about the project, the oral presentation that is done